Assalamu alaikum wa rahmatullahi wa barakatuh. Bismillahir Rahmanir Rahim. Welcome to the second class of biology of the chapter 14, Biotechnology. So, in the previous class, we have learned about some aspects of biotechnology. We have learned what is the concept of biotechnology, and we have discussed about the classical era and modern time era of biotechnology. And we've also learned about how plant tissue culture is performed in laboratory and what are the practical benefits of plant tissue culture process. And we've also learned some key terms from the previous lesson. This key terms was definition of biotechnology, and also uh, the another name of plant tissue culture is called micropropagation, and we have called some cells as totipotent stem cells. They has the capacity to convert into any kind of cell. Explant is the part of mother plant used for plant tissue culture purpose. Culture medium is the artificial jelly-like substance used for plant tissue culture process that provides nutrients and growth hormones. Callus is the undifferentiated cell mass produced from the explant in the culture medium. Sterilization is the process of killing bacteria and fungus present in culture medium and explant surface. And acclimatization is the process of adapting to the natural environment of a tissue culture grown plantlet. And today, the objectives are we will learn about the concept of genetic engineering. That has another synonym of recombinant DNA technology, and we will also can identify which organisms are called GMO, and transgenic organisms are a subset of GMO. And we can also learn the common steps followed in recombinant DNA technology or genetic engineering, and we'll also learn the role of bacterial plasmid and different enzymes like restriction enzyme and ligase enzyme used in tissue culture process, used in recombinant DNA technology, and use of recombinant DNA technology or organisms in agriculture, therapeutics, fisheries, and environment conservation. And along this lesson, we will find different key terms like GMO, transgenic, plasmid vector, restriction and ligase enzyme, transformation process, biosafety protocols, BT transgenic plants, PRSV, and interferon. So let's start with the concept of genetic engineering. So from the term genetic, we can assume that there is any kind of manipulation or that deals with genes present in an organism, and engineering means the manipulation. So genetic engineering is the technique of manipulating or transferring desired gene from one organism to desired organism. Actually, in broad sense, genetic engineering is called any kind of artificial man-made uh, technique or manipulation applied on genome of an organism so that the gene is changed, so the organism produces different characteristics so that we can find something beneficial or new from that organism. So genetic engineering is also called gene cloning because a copy of gene is made. So by the process of genetic engineering, we can done many things. We can delete a gene of an organism, or we can change the gene of the organism, or we can insert a foreign gene from somewhere to the organism. So when we transfer a foreign gene to an organism, so actually we clone or make a copy of the gene and paste it inside the organism. So this process, that is why, is also called gene cloning. And genetic engineering is also called synonymously recombinant DNA technology because the DNA of an organism is recombined or changed. So before genetic engineering, the organism has its own native DNA, the collection of genes, but when we genetically engineer it or modify it, it has a different set of gene than previous. So That is why DNA is recombined and called recombinant DNA technology, but in short, rDNA technology. And the organism having a new altered genome is called recombinant DNA. So let's start with the concept of GMO. What is called GMO? GMO stands for genetically modified organism. That means the organisms whose genome has been intentionally, artificially modified by humans by any kind of process. So we can apply different kind of methods, different kind of techniques to genetically modify an organism. So any kind of organism that has been artificially, intentionally genome changed, genome modified, are called genetically modified organism, or GMO in short. And among the GMO, a subset of GMO are called transgenic. So a subset of GMO organism are called transgenic, whose in which foreign gene has been transferred. So among all genetically modified organisms, there is a group of GMO which are also considered transgenic, because to them foreign gene has been transferred. That is why we say that all transgenic organisms belongs to GMO as a subset of GMO, but all GMO doesn't fall under the category of transgenic organisms. Now we will discuss in detail how recombinant DNA technology is applied on an organism. That means gene is transferred and the organism DNA is changed so that it produces new feature. So as an example of recombinant DNA technology, we can examine the production of recombinant DNA insulin inside a bacteria. So we know that insulin is an essential hormone for humans and for all mammals to metabolize the glucose properly inside the body. If there is lack of insulin, so that the glucose level will be high enough, will be persistently high enough inside the blood, so that the cells and different organs gets damaged, and also the body doesn't get the enough glucose inside the cells, because the glucose cannot penetrate inside the cells. So who are suffering from deficit of insulin, that the people who has permanently lose the capacity to produce insulin inside the islets of Langerhans of the pancreas, this person should be provided insulin hormone by injection in their body. So how they will get this insulin hormone? And the previous attempt was that for about 100 of years ago, about 50, 60 or 100 years ago, before the production of synthetic insulin, artificial insulin, people used to collect insulin from different mammals like pigs or goats or sheep, and they kill them and used to extract the insulin from their pancreas and isolate it and purify it, then made it available for human use. But this earlier tradition has some problems, because you have to kill a lot of pigs or sheep to collect only one vial of insulin hormone. So that is not actually practical. So scientists developed most probably in 1988, a company called Eli Lilly has first produced and commercially made it available, recombinant DNA insulin, and they made it inside a E. coli bacteria. So we know that an E. coli bacteria, it is not supposed to produce insulin, because it is not a feature to produce insulin inside an E. coli bacteria. So we see that there is a E. coli bacteria. So normally a E. coli bacteria doesn't produce any kind of insulin hormone, but we want to genetically modify it, and we want to transfer human insulin gene inside this E. coli bacteria, so that E. coli can produce insulin hormone, and that can be further used for human use. So for the production of insulin inside bacteria, we can apply the recombinant DNA technology. So when we start, At first, we have to extract the donor DNA and the plasmid. So what we want to produce inside the bacteria, in that case we want to produce insulin hormone inside E. coli bacteria. So we have to isolate the insulin gene from the human body cell. So we have collected a body cell from human, a somatic cell, and it has a nucleus, and inside the nucleus it has the long 2-meter long DNA, and this DNA contains approximately 30,000 genes, but we don't need all of them. We only need the insulin gene. So we have collected the DNA from human, and also we have collected the plasmid from E. coli bacteria. What is plasmid? Plasmid actually is a kind of circular extranuclear DNA segment found in some bacteria. For example, this E. coli bacteria has a main nuclear material that is, but inside, with it we notice a circular segment. There is actually a circular segment of DNA that is called extranuclear DNA. That is actually not essential for the E. coli bacteria, but it provides some genes which helps the E. coli bacteria to survive in adverse conditions. So if we can take out this plasmid from the E. coli bacteria, it will not get hurt, it will not die. So we can collect the plasmid from this E. coli bacteria, and you use it as a carrier of the human insulin gene. So we can collect the plasmid, and we can stick the human insulin gene with the plasmid, and again we can return the plasmid back to the E. coli bacteria. So that is our procedure. Number 2, we make fragments in the donor DNA and plasmid using suitable restriction enzyme. So what we do is that we don't need the 30,000 genes present in the human cell. We need only the insulin gene. So we have to make a fragment, make piece of the total 2-meter long DNA, and we have to cut only the insulin gene. So how that is cut in the particular segment? The desired gene is cut using specific suitable restriction enzyme. So we can collect the insulin gene from the rest of the genes, and we can also make a free end inside the plasmid, because we see that the plasmid is circular and there is no free end. So how can we stick an desired gene with it if there is no free end? So when we can cut this plasmid into some specific segment that makes it open space, so that I can stick the desired gene on the free space of the plasmid. So that is done by using suitable restriction enzyme. And then number 3, this process is called joining the desired DNA with fragmented plasmid by ligase enzyme. So what we do now is that we attach the insulin desired gene with the open-ended plasmid. So how we join this? We join these 2 segments using a specific ligase enzyme. The function of ligase enzyme is to attach 2 DNA segments. So now a plasmid has been created. This plasmid is not the original one. It has been changed. It has a different type of gene now. So that is why this plasmid is now called recombinant plasmid. And then this recombinant plasmid is transformed, that means inserted into the original bacteria. So now this plasmid carries the desired gene, insulin gene, and it has been returned back to the original bacteria, so that the original bacteria receives the recombinant plasmid, and now this plasmid will be called, this bacteria will be called transgenic bacteria, because a foreign gene has been transferred to it. And then we have to identify whether the bacteria has successfully received the transformed plasmid or not. So we have to make it sure that the bacteria has received the transgenic recombinant plasmid. So when we get confirmed of this phenomenon, when we identify that and we make appropriate conditions for the bacteria to propagate, because this single bacteria will produce a small amount of insulin hormone, because a bacteria is very much small and its production capacity is very much small. So that is not practically feasible to produce a large amount of insulin commercially from a single bacteria. So we have to culture and rear up these transformed transgenic bacteria. So what we do, we transfer this transgenic bacteria to suitable conditions in large tankers that have suitable temperature, suitable oxygen supply, and necessary nutrients, sugary materials, and growth materials, etc., so that it can multiply rapidly, so that it can produce billions of bacteria within several days. And when all of them produce their own enzymes or own hormones, own proteins, along with it they will also synthesize the insulin hormone. So we see that there is a tank in the and this tank produces genetically engineered insulin. So these tanks are called fermenter tanks. Inside this transformed insulin bearing bacteria are cultured, and they are kept in these fermenters for several weeks or months long. And after a specific time period, when a good amount of insulin hormone is produced, then the bacteria are killed, and the insulin protein is extracted by using different chemical processes, and then purified, and then that is marketed in small vials of insulin hormone. So if you have any diabetes patient in your home, then you might see there's some kind of vials in your refrigerator. That is actually a refrigerated recombinant DNA insulin for human use. So most probably you can see this insulin vials in your refrigerator at your home. That is actually called rDNA. That means this human insulin has been produced by recombinant DNA technology. So there are the processes followed in recombinant DNA technology. The first one is extraction of DNA and plasmid, then fragmentation of plasmid and human DNA, and then joining the insulin gene with plasmid, and then transforming the recombinant plasmid on bacteria, and then identifying the transformed bacteria, and then multiplying it in a specific culture medium, and then extracting the product from the culture medium and marketing it. Now we will compare some differences between conventional breeding and genetic engineering. So conventional breeding was used about several hundred years long ago. In that process we have made hybrids of different plants and animals. What we do, we take one variety of a plant and take another variety of a plant of the same plant and mix them together, and pollen from one variety of plant fertilizes the ovum of another variety of the same species of plant, and then new seeds are produced, new plants are produced which contain some features of the father plant and some features of the mother plant. So that was the process of conventional breeding. By this process we have developed different rice varieties or wheat varieties, and conventional breeding purpose was also prominent when you see the different varieties of dogs. As you see, there are different varieties and sizes and shapes of dogs we rear for domestic purpose, and actually there are numerous number of breeds of dogs. But how they came, 300 or 400 years ago, they were not they don't exist in earth. But how different breeds were created, different breeds of dogs were created by selective breeding procedure. By selective breeding programs, they have produced different breeds of dogs. So you have so that you can find small dogs that can be carried on a vanity bag, and you can also find some large dogs which are similar in size with a cattle, with a cow. So now we will compare the differences between conventional breeding and genetic engineering process. At first, by conventional breeding process, gene can be transferred to closely related species. You can transfer gene by natural mating process or by pollination to the same species, or to some extent closely related species. But you cannot transfer gene, transfer pollen, or transfer sperm to a distant organism, distant species. That is not possible, because at that case the pollen cell or the sperm will get rejected uh, by the by the other species, by the others, or by the ovum of other species. They reject it when they see that the chromosome number and the structure doesn't make compatible. They reject it. So it is not possible. But in genetic engineering, you can transfer gene to a completely unrelated species. For example, you can transfer the luciferase gene of a firefly that makes the firefly to glow at the dark, and you can transfer it to a tobacco plant, so that this tobacco can produce a dim green light at the night. Or you can transfer this firefly luciferase gene into a zebrafish, so that the zebrafish will glow at the dark. So this a weird phenomenon is possible in genetic engineering process. And in conventional breeding, we can the particular transfer particular gene is uncertain. We are not sure which gene will be transferred and which gene will be expressed in the offspring. We are very much uncertain about this. But in genetic engineering, we can transfer a particular gene, and we can be 100% sure that the transfer gene will be expressed or will be transferred and expressed in the transgenic organism. And in conventional breeding purpose, to develop a breed, to develop a variety, we need a long amount of time, several decades of research, several decades of time to develop a new breed uh, or new variety. But in case of genetic engineering, we can accomplish the transfer of gene within several years, within 4, 5, 6 years. That is very much fast process, and we can perform it within very short amount of time. And in conventional breeding, there is a benefit, that is, biosafety protocols and toxicity tests are not required to follow. That means in conventional breeding process, there is no risk of producing unwanted proteins or toxic proteins in the plant. So we don't need to follow these two protocols. That means biosafety protocols and toxicity test. That means whether the organism produces any kind of toxic protein or not, or if the organism is safe for the environment or ecosystem. But in genetic engineering, as we are manipulating the genome of an organism intentionally and artificially, we have to follow these two protocols, biosafety protocols and toxicity tests, because we have to evaluate the performance of this organism, and we have to evaluate the impact of this GMO organism on the ecosystem, on the natural environment, whether that is producing any harmful effect or not. And we have to also assess the toxicity test. That means if any kind of undesired or toxic genes has been transferred to the organism or not. So if the organism is producing any kind of toxic protein or not, we have to make sure about it. So these protocols have to be maintained in genetic engineering process. Now we will learn about some uses of genetic engineering in case of plant development, that is in agriculture. So by genetic engineering methods, scientists have developed some varieties of corn or cotton or soybean or rice, which are called BT corn, BT cotton, BT soybean, BT rice, etc. So why they are called BT and why they are also called transgenic? Because these organisms are genetically modified organisms. At first, we first see an example and comparison between BT corn versus non-GMO corn. As BT corn is a GMO corn and that is non-GMO corn, we find some differences. That is, the BT corn, the upper one, is healthy and it is not attacked, it is not infected by pests. Whether the bottom corn, non-GMO corn, is infected by pests and has been attacked. So that means BT corn is not susceptible to pest attack because it can survive the infestation of pests. How? This BT corn is resistant to the attack of Coleoptera and Lepidoptera orders of pest. This BT corn is resistant to the insects belonging to Coleoptera order and Lepidoptera order. So there's two prominent insects attack on corn plants and make it weaker and damages the plant, but they cannot attack on the BT corn. So why they cannot attack on BT corn? Because BT corn is a transgenic or GMO organism, GMO plant, which is produced by transferring toxic crystal protein genes from the Bacillus thuringiensis bacteria. That means crystal toxic protein gene has been isolated from a bacteria called Bacillus thuringiensis bacteria, and it has been inserted to a corn plant so that the corn plant became transgenic. That is called BT corn plant. So this is the bacteria and there is the colony of this Bacillus thuringiensis bacteria. So how this corn resists the infection of pests? So we have talked about it earlier that BT corn or BT cotton has crystal protein gene inserted that is collected from Bacillus thuringiensis bacteria. So in the whole body of the corn plant, toxic protein is present. Toxic crystal protein is present throughout the body of the BT corn or BT cotton plant. So what happens if a Lepidoptera or Coleoptera insect comes and eats a small amount of leaf or shoot or corn, then it ingests the plant part and the crystal protein enters into its midgut and becomes activated, and this crystal protein gene ultimately kills and damages the cell of the midgut of the insect, so that eventually within some days the pest dies. So that is why if some kind of pest invades in the field of corn, of BT corn, within some days they will get killed and they have to flee away from the field. That is how the BT plant works. Now there is a safety concern about humans. That is, if a small insect consumes it, then its midgut gets damaged and it dies. So what will happen if we eat this BT corn as our food? Is there any possibility of harm in our body when we consume them? The designers, the scientists who have developed it, this company says that it is not toxic for human consumption. That is toxic for the pests but not toxic for the human consumption. But there are some many safety concerns coming from some environmentalists and some group of researchers and scientists who are saying that it needs a long-term research to evaluate. A long period of research is needed to evaluate whether it has any kind of toxic impact on human body for consumption. We have to assess it by a long period of study. So there is an ongoing debate between in favor of this BT corn and in opposition of this BT corn. And now we will also learn about a papaya variety that is called PRSV resistant papaya variety. So the full form of PRSV is called papaya ring spot virus, and some kind of so when this virus infests or attacks the plant looks like this. So that is the papaya that has been infected with papaya ring spot virus. You see there's some green ring spots on the papaya. So by this symptom, the virus has been named as papaya ring spot virus. So when this virus attacks, it kills the plants and it reduces the yield of papaya at very much large extent. So scientists have developed a variety of papaya by genetic engineering, which is resistant to the attack of PRSV. That is one use. And I want to show you another use of genetic engineering to make tomato, that is called genetically engineered tomato, that has longer shelf life. That is not an example from your book, that is outside of your book, but I am interested to show that. This variety of papaya is called Endless Summer, produced in 1994. And this tomato has a unique feature that it can be stored and preserved in shelf or in store for a long amount of time, or compared to the conventional breeds of tomato. That means these tomatoes can be collected at ripened condition, and they can be stored for a comparatively longer amount of time in the store or in the shelf, and within this time the tomato will not rot or will not get damaged, because this tomato contains a bioengineered SSC synthase gene, which gene product ultimately inhibits ethylene production inside the tomato. So we know that ethylene is a growth hormone that enhances the ripening of fruit. So if there is less amount of ethylene production, the fruit will ripen very slowly, so that it can be preserved in shelf or store for a long period of time without any kind of preservation method. So that is why this genetically engineered tomato has a greater advantage, having longer shelf life. And now we will discuss about some herbicide resistant plants made by genetic engineering method. In US and most of the European countries, there is a strong herbicide used in the field. There is a use of strong herbicide chemical used in USA and European countries, which is called glyphosate. Sorry. So this glyphosate chemical is very much strong enough to kill most of the herbs or weeds present inside the field. But this glyphosate, its commercial name is called Roundup. So this Roundup or glyphosate is very much strong, that it kills all of the weeds, but it also kills the crops also. So is there any benefit? There is no benefit actually. When I want to get rid of the weed, but it is also killing the plant, so this is actually damaging my plant. So what to do? At this situation, scientists have came up with genetic engineering to genetically modify the crop plants, so that these plants can survive or withstand this strong glyphosate chemical. So that what happens when you apply the herbicide on conventional sugar beet, there is no weed, you see, but the growth of the sugar beet is also hampered. But when we use the sugar beet that is genetically engineered, which are Roundup ready, which is resistant to Roundup chemical, so when we apply this herbicide, the herbs get damaged and gets killed, but also the sugar beet plants, they can withstand this chemical application, so that you see the healthy sugar beet present in the field, and there is no weed also. So here you see what happens. In the left side you see that, in the right side you see the soybean field infested with herbs, and when we apply herbicide, the weeds are killed, and the soybean plants remain. So that is how the Westerners are killing the weeds, making strong chemicals to kill weed. And when they see that the crops are dying, so then they develop glyphosate resistant plant variety, that is GMO sugar beet or GMO soybean, etc. And now we will also learn about how genetic engineering increases the food value or nutrition value of crops. For example, you see that the first one, the white rice, is the conventional rice we eat, but at the back you see that there is yellow colored rice. That is actually genetically modified rice variety, which is called golden rice because it is white in color. But to make it, because that is golden in color, but to make golden in color is not the objective. This golden colored rice contains very much high amount of vitamin A compared with the conventional varieties of rice. Scientists came up with this plan because in the developing countries most of the people eat rice, but they also suffer from vitamin A. They suffer from the deficiency of vitamin A. So what to do for these people? So some scientists came up with an idea that if we can incorporate a gene for synthesizing vitamin A inside the rice, so that people will get their nutrition demand of vitamin A by simply eating the rice alone. So from this idea they have developed this genetically modified golden rice that produces high amount of vitamin A. And now we will also describe about some genetically modified animals. Some genetically modified animals are used for therapeutic purpose. For example, this cow is called Rosie, that was created in 1997, and this cow is genetically engineered so that it produces a human protein, that is alpha-lactalbumin, inside its milk. Because the cow milk doesn't contain the alpha-lactalbumin protein, which is present in human milk. So how to make the cow milk closer to human milk in quality? So they have introduced the alpha-lactalbumin gene collected from the human and inserted it into the cow, so that inside the cow's milk we can find the protein, alpha-lactalbumin protein, which has a different valuable therapeutic purpose. And also we can see a genetically engineered sheep. You see the right one is bigger in size than the left one, because here you see the right one is bigger in size than the left one, because the right one is transgenic sheep. It has a extra copy of growth hormone gene, so that it produces excessive amount of growth hormone, so that it grows bigger at the same amount of time compared to the normal sheep. And in your book there are also two examples of genetically engineered animals, that is scientists have inserted cis-E and cis-M gene from bacteria and transferred it into the sheep, so that the sheep has a good quality of fur, wool. So how they did it, why they did it? Because the bacteria contains cis-E and cis-M protein. These genes actually are responsible for producing cysteine amino acid inside the body. So when cis-M and cis-E gene are inserted into the sheep, then the sheep now can produce enhanced amount of cysteine protein, so that this enhanced amount of cysteine protein can contribute to better quality of wool in its. And now we will discuss about production of a human growth hormone, that is recombinant DNA, that is produced by recombinant DNA technology. So we know that human growth hormone is essential for proper human growth in length, that has and and that can be produced in bacteria or in yeast by uh, genetically modified process. So here you see some vials, which vials contain human growth hormone recombinant DNA origin that can be administered on patients who suffer from deficiency of it. You know that the football player Messi uh, has he suffered from deficiency of human growth hormone when he was a teenager. So so he was very much shorter in size, and now you see. So what happened to him? The club who hired him, they offered uh, all the expense for his treatment, and offered the regular treatment, regular administration of human growth hormone, so that higher in length for several inches after this administration of human growth hormone. And then we are discussing about use of interferons. We say that interferons are some protein chemicals used to treat leukemia, hepatitis B or C, or any or other viral diseases. And interferons are naturally produced inside our body, but when these cancer conditions or viral infections trigger in the body, we have to block the growth of virus or cancer cells, and then we have to administer interferon from the outside into the body. So how we can produce interferons outside the body? Interferons can be produced inside yeast or bacteria by genetic engineering processes, and they are used for treatment. You see the vials of interferon. Actually, they are very much expensive vials, chemicals, and they are administered in cancer patients or viral infected patients to boost their immune system, to suppress the cancer or virus in their body. And now we will discuss about the role of genetic engineering in fisheries.
Exclusive look at some.
So here we'll see the production of genetically engineered salmon fish. Salmon fish is a very much famous and tasty fish, and popular among the Europeans or Western people. And this salmon fish is actually comes from sea, and it has a very much low growth rate. So scientists have manipulated it to grow it bigger in size. So what they did, they have engineered and they have inserted some growth hormone genes inside the genetically engineered inside the salmon fish, so that it becomes genetically engineered and it can get larger in size, almost twice in size than the conventional salmon fish. So here you see the video.
Thing that may be approved by the government as safe.
So there is a TV report that shows about the genetically engineered salmon fishes. But there are some environmentalist people who are opposing these genetically engineered salmon fishes. They are fearing of different damages or bad consequences from it. So they are salmon fishes, and this farm produces genetically engineered salmon fish. And here you see that there are two fishes. The upper one is the normal salmon fish, normal breed of salmon fish, and the bottom one, larger in size, that is the genetically engineered salmon fish. That is almost twice in size of the normal breed. So that is the normal fish, and that is the genetically engineered salmon fish of the same age. So that is done by genetically engineering processes, by incorporating growth hormone genes from two other different fishes, so that the salmon became genetically engineered. So that's how genetic engineering has been used to increase the growth rate of fishes in salmon or nilotica or tilapia, etc. And then at the last, for the environment protection, genetic engineering can have some roles for us. For example, they can help in bioremediation. That means to uh, make the damaged environment into normal condition. So we can remediate, or uh, we can compensate, or we can minimize the damages done on the environment by using genetically engineered organisms or bacteria. For example, as an example in your book, you see you see the name of Ananda Mohan Chakraborty, and he is actually an Indian American microbiologist who genetically engineered a bacterial species that is called Pseudomonas putida, and that can degrade oil spills on water or soil, and he discovered it into 1971. So what happens, what is the use of it? And there is a bacteria strain, and what he did, he actually genetically modified a Pseudomonas bacteria strain and named it Pseudomonas putida, and that has an excellent capacity to degrade oil uh, floating on water or soil surface, because this bacteria has unique capability to degrade oil or complex hydrocarbon materials, and they get energy by degrading them, and they can multiplicate at this condition, and while other bacteria don't have such property. So this bacteria can be used at different wastelands or damaged sites or oil spill regions, etc. So today we have learned about what is called genetic engineering or recombinant DNA technology, and we have also learned what are called GMO and what are called transgenic, and there is a sharp difference between them. And what are the common steps followed in recombinant DNA technology? For example, how to make a recombinant DNA insulin by recombinant DNA technology process. And we have also learned the role of bacterial plasmid registration and ligase enzyme in genetic engineering, and we've also learned about the use of different recombinant organisms in agriculture for increased yield, or for make them free from pest, and also use in therapeutic purpose, or fisheries, or for environment conservation. And we've also learned different key terms, and that is transgenic, GMO, plasmid vector, restrictional ligase enzyme transformation, biosafety protocols, and BT transgenic plants, PRSV, and interferon. So at last you have a homework that describe the procedures of recombinant DNA technology. No more today. সবাই ভালো থেকো ইনশাআল্লাহ। আসসালামু আলাইকুম।